and welcome to 2024. I'm pleased to announce today's speaker. Oh, my name's Andrew Muir, by the way, Great Lakes Fishery Commission. And I'm pleased to announce today's speaker to kick off the 2024 webinar series, Dr. Bo Bunnell. Bo earned a Bachelor of Science from Center College in Biology, a Master's from Clemson University in Fisheries, and a PhD from Ohio State in Evolution, Ecology, and Organismal Biology under Dr. Roy Stein. He conducted a couple of postdocs, one with Tim Johnson at the Ontario Ministry of Natural Resources in Ontario, and a second with Tom Miller at the Chesapeake Biological Station. Bo has been a USGS research fish biologist since 2004, where he worked in the Deepwater Science Program until 2020, when he took on the role of coregonine Science Program Director under a long-standing partnership between the U.S. Geological Survey and the Great Lakes Fishery Commission. Today's presentation was given as a plenary at the last Coregonine Symposium in France within the Stock Assessment, Fishery Management, Conservation, and Aquaculture session. Bo, we look forward to your presentation, and take it away. We can see your slides here perfectly.
Okay.
All right. And just one more thing, as always, we are recording and we will be posting this on our YouTube page as well after this is finished. So this as a consent reminder for the recording.
Thank you, Renee, for all you do to facilitate and host, and Andrew for that introduction. And we definitely appreciate the partnership and support from the Great Lakes Fishery Commission for our coregonine restoration framework. All right, so you get me today talking in a different way. This is what happens when we scramble and can't find a volunteer for January. So I will recycle or share potentially to some of you that haven't seen it before, a talk that I gave over in Evian, France back in September. And as Andrew noted, it was a relatively broad session that day. Stock assessment, restoration. And I was sort of struggle with how I would go about providing a sort of an overview talk that might be helpful to that group of us that were convening at the Coregonine Symposium, but that also might help what we are doing here with Great Lakes restoration and conservation. So I decided to undertake a literature review to try to determine what management actions have been taken, as best we can tell, to restore or conserve coregonines when those ciscos or whitefishes are the target species of interest. And as I got into it, and you see that I was able to persuade several people outside of the US to help me find all the evidence and sort of case studies that we could. And so I'll start out with sort of a broad introduction as to why we need to potentially restore or conserve coregonines and some of the threats and stressors that they have sort of had to deal with over the past century or so. The first one that, again, is common across both continents, all three continents really, for this circumpolar distributed family of coregonines is eutrophication and hypoxia. So our Coregonine Symposium was on the shores of Lake Geneva, as you see that image there, with a pretty rare but impressive cyanobacteria bloom shared by one of our co-authors, Orlan Anneville. And also you can see some blue-green algae blooms there in Lake Constance, another lake in Europe that's very well studied for coregonines. Invasive species we know have been a tremendous stressor in the Great Lakes. For coregonines, we know that sea lamprey probably had a potentially compounding effect with other stressors for some of the deepwater ciscos in the middle 20th century. Alewife have been hypothesized to be an additional stressor potentially through competitive or predatory effects on the larvae of some coregonines. Three-spine stickleback is a new stressor that has been identified in Lake Constance that is implicated in the decline of the lake whitefish abundance and diversity there. Overfishing, the best examples of this, of course, are in the Great Lakes. some images here, iconic images from Bayfield, Wisconsin, Lake Superior. And this is from the Eschenroder et al. Compendium that was published in 2016. Of course, that iconic image of the Earl Bess with Cisco overflowing from it in Lake Erie. Habitat degradation. This is one where we can sort of start at the watershed and look at how land use changes have affected nutrient inputs or sedimentation to the lakes. And that could be sort of point sources, as you see there in the bottom left, or it could be larger scale efforts like along the riparian zone in many of the Great Lakes, where we essentially deforested the habitat, would float the logs down the river down to the sawmill and lead to tremendous large inputs of sedimentation that often would fill in those key interstitial spaces that are important for salmonids and for coregonines egg incubation. And then lastly, their channelization. This is the dewatering of the Detroit River, which happened more than a century ago for the purposes of navigation, but obviously had big impacts on fish spawning habitat, including spawning habitat of coregonines. Climate change is a relatively new one, and we can think about this in multiple ways. Some of the bigger ones are in terms of changes in precipitation, leading to drought years, as shown here. And this is Lake Constance in a drought year where some of that nearshore habitat is no longer accessible to fishes. Temperature reflected in water temperature, and when ice occurs also has been changing. These are, you're seeing here the date of the first freeze for many lakes through a compilation provided by EPA. And then an example, again, in Lake Constance provided by Ole Seltz, showing that the start of the spawning of lake whitefish or European whitefish in Lake Constance has been increasing, or going later and later as average November temperature has been increasing. The last one is one that is not always a stressor, but sometimes can be a stressor, and that's stocking fish. So stocking can have effects, as I'll talk about later, not only on the potentially through domestication and integration and hybridization on sort of the species that you're trying to conserve, but it also can have negative impacts on some of the native species. And one sort of interesting study that Christy and all put out showing that even in cases where we're doing a really good job raising fish and sort of releasing them responsibly, as we typically do in the Great Lakes, their relative reproductive success typically is lower than their wild counterparts. So they just aren't the same as reproductively successful as the wild fish. So with all of those challenges, it's not surprising, and it wasn't surprising to this audience here or to the audience at the Coregonine Symposium, that we've seen tremendous losses in biodiversity. We've seen extinctions, sadly, some local extirpations, and then many species that are threatened and in need of conservation. This is an illustration that Andrew provided. These are actually illustrations from Paul Vecchi that were beautifully provided in the Eschenroder et al. Document, but just described the wonderful Cisco diversity that we had in the Great Lakes. And some of these are sort of sub-specific diversity of some of our Cisco species. But you can see, especially in Michigan and Huron, tremendous diversity of Ciscos that once occurred. And sadly, we've lost quite a bit of that. In fact, we have places like Lake Erie without any Ciscos anymore. Lake Ontario with just one of its original Cisco species. Lake Superior still is intact, but I think if you were to talk to people like Owen Gorman or Amanda Ackes, they would tell you, or Tom Pratt, they would tell you that we're still trying to understand the diversity, existing diversity that we have in Lake Superior today. So what does that look like? If you just sort of take all of the species across all the lakes and what we lost, to at least 70% of that diversity was lost in the Laurentian Great Lakes. 3 species are extinct, assessed by either the US or by Canada. And 4 species have had sometimes multiple extirpations from multiple lakes. So it does provide, because some of these species are still extant in some of the, in Lake Superior or in Lake Michigan or Huron in some instances, it does provide an opportunity for a reintroduction or restoration in the lakes where they have become extirpated. There's a similar story in Europe. One of the best examples is in Switzerland. Many lakes, some of the deeper lakes there, including sort of Lake Geneva shown here. Again, we were on the sort of the France side of Lake Geneva, but there are 24 different extant whitefish species described in Switzerland. Now they do their taxonomy based more on local diversity because they feel like that's the best way to protect that diversity. So it's perhaps a little bit different way than we sort of describe taxonomy now, sort of in the US and Canada. But even with that sort of more splitting, if you will, of the taxonomy descriptions, 10 of those species have been lost, are now extinct. And there have been 4 local extirpations. And the biggest stressor, and this is well summarized in Pascal von Lanthen's 2012 paper. The really cool thing in this paper is they're able to attribute those declines with eutrophication. And so here in this, I pulled this out of their paper, it's figure or the panel D. You can see increasing levels of phosphorus or phosphate up through about 1980 across multiple lakes, and then a subsequent oligotrophication down to levels, less than 10 in some cases, or 20 or 30 in other cases. And they've been able to sort of describe or relate the peak levels of phosphorus are positively related to the highest, like 100% of those species losses in some cases. And lakes that didn't achieve the highest levels of phosphorus had lower levels or lower proportions of those species that were lost. It's not just the species that are lost, and this is another sort of interesting way of thinking about it. It's also the functional diversity of those species. So if you look here in the variation in a measure like gill rakers, which we of course attribute to variation in sort of trophic ecology or different sorts of prey types, the lakes that have still the highest diversity in terms of gill rakers, including short, few gill rakers and longer, higher number gill rakers, those still exist in areas where the intensity of the past eutrophication was relatively low. And as you go to places like Lake Zug, where the eutrophication was super intense, like beyond what we, this is like Western Lake Erie on steroids, very low variation in the gill rakers that still exist today because of the loss in that diversity. So diversity, of course, matters in terms of the resiliency and the connectivity or the complexity of that food web. But all that negativity, there really is some reason for optimism. First of all, as you saw there from those Switzerland lakes, also in North America, we have seen nutrient input controls for the Great Lakes Water Quality Agreement, for example, that have led to oligotrophication. We also have greater connectivity where we're working to create more fish passageways or outright remove dams. Fishing mortality is obviously much more regulated than what it was 100 years ago. And when we lost so much of that diversity in the Great Lakes. And then in terms of invasive species, some have been controlled. So in a case like sea lamprey, or some are not as successful or as abundant as they used to be now that we have a more changing environment. And so I think sort of we can say that we are now potentially embarking on an era of coregonine conservation or restoration, and not just a period of sort of watching the losses of the diversity that we endured over the past, but in the early 20th century. So as I get into this talk, then I want to just make 2 sort of quick definitions. Conservation, of course, would be And this talk covers both conservation and restoration. Conservation would be protecting existing species and their critical habitats. And it often is mandated through legislative authorities to lead to action. And that's when that was the case, that's when we found the best examples in the case studies that we reviewed. Restoration, defined by the Society for Ecological Restoration, is defined as assisting the recovery of the ecosystem that's been damaged, degraded, or destroyed. And this can take multiple forms. It could talk about restoring that key habitat, removing invasive species, or reintroducing native species that have been extirpated. So in order to try to get a better sense of what has been done with respect to conservation and restoration of coregonines, I did a traditional literature review with more than 40 different search terms and combinations, showing you some of the search terms there. Other ones were things like whitefishes or ciscos, some of the common names as well. Using Web of Science, using Google Scholar. And then the second thing I did more intensively was looked at all of the different previous coregonine symposia proceedings. So these are, you know, our library here at the Great Lakes Science Center has a really nice archive that I could carefully comb through. These date back to 1970 and have occurred more or less every 3 years since they were revived in the early 1980s. But the thing that really allowed me to get the best insight was to consult these regional experts. And I was really grateful that almost everybody I reached out to ask if they could help me in this endeavor kindly said yes. And so these are the names of those individuals. They come from France and Germany and the United Kingdom and Finland and Sweden and Switzerland and Austria. And then Weidel, I reached out to just because I knew that he has done a lot of literature review on restoring habitat, given his leading work in coregonine habitat in the Great Lakes. So to do this sort of remotely, we used Google Sheets as a way of compiling not only primary peer-reviewed literature, but case studies that were documented only in the sort of the white papers of the gray literature. And so I'm going to just show you a few high-level results, and then I'll talk about, from there, I'll talk about how I'm organizing the rest of this talk based on different categories of restoration and conservation. So the first thing one of the first sort of take-homes is just focusing on those coregonine symposia. And since 1995, all of the plenary talks and then all of the presentations themselves have been organized into different themes, if you will. And if you take those theme names upon which the papers or the presentations are organized, you come up with 36 words or key phrases, you know, since 1995 that sort of give you a flavor of what the primary focus of these coregonine symposia have been. And so their word cloud, and the word cloud comes out like this, where you do see conservation showing up there in green, but in fact, it's the 9th most common of the words or key phrases. And they really don't start showing up until 2008. And restoration never showed up at all. And so the other way of looking at this, again, this is a high level sort of result I'm giving you first, is to look at the number of water bodies in which these different categories or different restoration or conservation actions have occurred. And so this is, of course, very imperfect because we know we didn't sort of capture every different conservation or restoration action that's targeted at coregonine species. And, you know, if you were to be doing this, you might organize these different conservation or restoration actions in different groupings, perhaps. But the way we did it was we grouped them into the stocking of hatchery fish, the controlling of invasive species for the benefit of coregonines, translocation of fish, restoring habitat for the benefit of ciscos or whitefish, and then finally creating protected areas where fishing mortality cannot be occurring. Or another way of saying protected areas would be just to eliminate fishing altogether. Not just in one area, but from an entire lake. So when you look at the sort of the spread of what those look like, this may be not too surprising to you that stocking was sort of the most common result. I think stocking is a common and effective tool for managers in the Great Lakes, but this also showed up across this global sort of effort. And so what I'll do now is sort of unpack, share with you some case studies of these different categories. And where I, of course, really learned a lot is learning how people have used some of the less common strategies, translocation, habitat, and creation of refuges or protected areas. And so that's like the next 20 or 25 minutes of the talk. So let's jump right into it. So stocking of hatchery-reared whitefish and cisco actually dates back a long time in Europe and in North America. However, the primary purpose has been the creation or supplementation of fisheries, which is, of course, outside the scope of this talk, where the focus is on the conservation of imperiled species or the restoration of extirpated species. As I mentioned earlier, one of our co-authors provided a study that shows that coregonine stocking in Norway can lead to strong negative effects on other native species. And then even from a conservation perspective, we know, especially from work out in the Pacific Northwest with some of the salmon stocking that has occurred, hatchery rearing can lead to unintended negative effects on congeners or conspecifics. And so this can occur through selection of undesirable traits or domestication, and then bringing those traits into the wild where they can be, those animals can hybridize and introgress back with conspecifics or potentially with congeners. Of course, also the introduction of disease. However, the stocking of hatchery origin fish for reintroduction is obviously one of our key management tools. If the previous threats that led to the local extirpation have been removed and population viability is believed to have some reasonable probability of success, reintroduction of hatchery origin fish certainly is a viable restoration strategy. So the first example I'll share is in the Rhine River in Germany, where Houting, or Coregonus oxyrhynchus, was extirpated in the late 20th century due to overfishing and habitat degradation. So managers used Houting from the river Vida in Denmark as gamete sources, introduced those fish back into the Rhine in the late 1990s and early 2000s. And since that time, they have detected natural reproduction of those Houting, now back restored in the Rhine River. The only sort of asterisk there is because there's a little bit of a scientific controversy as to whether Houting in the River Vida is the same species as Houting that was extirpated from the Rhine River. And that's based on morphological differences. Genetic information, however, is lacking to confirm that hypothesis. So coregonines are just as problematic in Europe as they are in North America. Another interesting example of the stocking of hatchery origin fish for reintroduction is for Prosopium species, round whitefish in the Adirondack region of New York. And so round whitefish are actually threatened in New York State. They were once distributed in more than 80 lakes or ponds in the Adirondacks, but have been extirpated in more than 75 of them, owing mostly to invasive species, mostly through predation, some competition, but also habitat changes through acid rain. So New York State has undergone targeted reintroduction in 24 of those 75 lakes that seem to be most amenable to success. But so far, I've only documented natural reproduction in 4 of those to date. But that's another nice example of using hatchery root fish for reintroduction. The next 2 you're going to potentially be even more familiar with because it's not just in North America, but in our Great Lakes. The first has been going on since 2012, where coregonines hoyi or bloater from Lake Michigan have been reintroduced into Lake Ontario, where they were extirpated by the 1970s. So New York State DEC, Ontario Ministry of Natural Resources, U.S. Fish and Wildlife, and USGS began reintroducing juvenile bloater back in 2012. And over those 11 years, about 1.3 million animals have been stocked back into Lake Ontario. So far, there have been about 14 recaptured fish. And Brian Weidel has done a sort of a back of the envelope exercise of estimating how many animals there may have been swimming in Lake Ontario in 2019 based on some estimated survival rates and how many fish have been stocked. And the estimate is tens of thousands potentially are now swimming in Lake Ontario based on this reintroduction effort, multi-agency reintroduction effort. The second example is from Lake Huron, Saginaw Bay. Cisco in Saginaw Bay really provided the heartbeat of the fishery and that population of typical arteti in Lake Huron historically. By the 1960s, however, they were extirpated. And so a reintroduction was prioritized by the fishery managers, and the Fish and Wildlife Service began reintroducing Cisco into Saginaw Bay in 2018 through great effort of many people in that agency. And so since 2018, more than 4 million juveniles have been stocked. So in only 5 years. So many more fish per year than what we've been able to rear for bloater. And there's some strong evidence of success with more than 50 adults being recaptured during evaluation efforts during the spawning period in December of 2022 and 2023. And those are data provided by Jose Bonilla Gomez of Fish and Wildlife Service in Alpena. But this, the restoration and the reintroduction story in Lake Huron is a nice illustration of some of those conservation concerns and how managers can balance those. And so those eggs, the source of those gametes to bring them back into Saginaw Bay, there were a couple of different sources considered both within the lake and sort of outside the lake. But the decision was made to be more conservative to protect those existing stocks from potential integration. And so they decided to use Cisco from northern Lake Huron rather than Cisco from outside of Lake Huron to prevent that potential outbreeding depression of that locally adapted Cisco stock in Lake Huron that has persisted even when it failed in Saginaw Bay. So the second strategy is one I admittedly knew less about, translocation. And so translocation, some of the best examples are actually outside of fisheries. And one really nice example is the gray wolf in Yellowstone National Park. And so by the 1920s or so, gray wolves were extirpated from Yellowstone National Park. By the 1990s, the National Park Service and several other Department of Interior agencies worked together to bring gray wolves back. And you see an image here of the first 8 gray wolves being translocated from Alberta, Canada, down into Yellowstone, where they were held in pens to help them sort of imprint for their first several weeks. And that translocation was successful. You see there on the bottom right, the relative stability of that population within the park. And so for fishes, when we think about translocation, this is essentially moving fish at any life stage directly between water bodies or minimizing the time in artificial rearing. So that is only incubating the embryos in the hatchery, which is how we are defining it, to try to reduce those domestication effects. Translocation, however, does pose risks. You have the potential movement of pathogens or parasites. You might have too few fish that are being translocated, which could reduce genetic diversity and lead to sort of founding effects where you don't have enough genetic diversity, and could lead to bottlenecks down the road. There's not many instances of translocation with coregonines, and most of our examples actually come from the United Kingdom with Coregonus laberatus, or the European whitefish, and with Coregonus albula or Vendace. And all of these in England, Scotland, and Wales were due to essentially legislative mandated efforts to conserve these species that were very rare on the UK islands there. So first, just talking about European whitefish, their goal here was to use translocations to create refuge populations, because the very few populations where the European whitefish still were living were very stressed and their numbers were declining. So there were, in Scotland, there's only 2 water bodies that European whitefish. The first was in Loch Lomond in the late 1980s, early 1990s. They translocated 15,000 unfed larvae and 100 adults to 2 nearby water bodies, and that was successful. And just through that translocation effort. And by 2005 and 2006, they had sampled wild reproduced whitefish in both of those new refuge lakes. However, they noted that they were morphologically different than what they were in Loch Lomond. The second example where they were sort of in Scotland, where whitefish was in Loch Eck. Also in a very stressed population there. So they selected 4 new water bodies to translocate embryos and unfed larvae from 2009 to 2011. Once again, there was success in that translocation, even translocating these earlier life stages than what we might think about as possible in the Great Lakes. But they did find that those animals not only were different morphologically, but also were different genetically. So some evidence of genetic drift. So those founding effect that I referred to as a possibility earlier. And they also were functioning a little bit differently. Their ecological niche was different than what it was in Loch Eck when they were in this new environment. And so they had some recommendations about making sure that at least 100 genetically diverse animals are translocated if you're going to be using this strategy. There's also a couple of examples for Vendace, as I mentioned. Sadly, Vendace were extirpated from the 2 original water bodies in Scotland where they occurred. This was by the 1970s, but they were still hanging on in 2 English water bodies. The first was Bassenthwaite Lake, where Vendace were in trouble, but still hanging on. So they, again, created these refuges. The first was to move them into 2 Scottish water bodies, but that did not work. So as they tried again in 2 additional water bodies in 1997 and 1999, and one of those, they actually were successful in creating a Vendace population in Loch Skene. The end of that story is a little bit sad. However, Vendace ended up being extirpated from Bassenthwaite Lake, but they do live on in Loch Skene. Before we sort of end the translocation discussion, I just want to note that translocations are common also to create fisheries, especially in Finland. So again, it's another place we can look to sort of, if we wanted to bring this to the Great Lakes, to learn how best to sort of undertake this method. And then in Switzerland, translocations have their own history, relatively common. up through the first part of the 20th century. But in 1991, the practice was completely banned because they realized that translocation of fish was leading to declines in genetic diversity through that hybridization and introgression that I spoke about earlier, that they felt like was doing more damage because many of these translocations were for the supplementation of fisheries rather than for conservation purposes. And then finally, I'll just end by talking about, again, most of this translocation effort was done in Scotland and in the UK. And Ian Winfield was really the one that published the most about this. Ian is now retired, but has been really helpful just through emails and me learning more about this practice. And they published in the 2012 paper, the reason why they focused more on translocation than hatcheries, because they felt like the use of hatcheries completely or limited. Let me start again. They avoided, the use of hatcheries completely, or they limited their use to development only to the eyed egg or swim-up larval stage to guard against any inadvertent but significant genetic selection, which is almost inevitable if older life stages are retained in captivity. So that's potentially a little bit of a strong potential point of view biased against hatcheries. And I still believe, just editorially for myself, that hatcheries still can be of necessity and some great value in the Great Lakes, but it does provide a little bit of a window into their thinking of why they use translocation. And as I sort of wrap up, one of my hopes is that we might begin to think more about translocation in the Great Lakes as an additional tool for restoration strategies. Habitat. All right, so habitat, one of our sort of baseline assumptions in our Great Lakes restoration effort has been that there's a connection between habitat complexity and diversity. And this is true also for coregonines. So the greater habitat you have, the greater niches that are available for animals to occupy and potentially to evolve into. And so we see this, for example, in coregonines, where the habitat has been split up by different deep water ciscos occupying different spawning depths and times of the year to maintain that reproductive isolation. And they saw a similar thing in some of the Swiss and German lakes described by Pascal von Lanthen. Given that destruction of habitat can be relatively visible, I think it's kind of surprising that we didn't document more instances of improving it for coregonine conservation or restoration. We only found 11 water bodies of habitat restoration targeting coregonines. At the same time, we acknowledge that coregonines are probably benefiting from broader fish habitat restoration efforts. And more broadly, there seem to be a lot of efforts targeting habitat improvement for trout and salmon across Europe and North America, and that we think that coregonines are definitely a beneficiary of those fish habitat restoration efforts, given their common spawning habitat that they can share. And then I wanted to note this really interesting study that was published last year by Radinger et al. In 2023, where they undertook a relatively large before-after control impact study across 20 German lakes, 6 years, where they did one of three things. They either created more shallow littoral zone, they added coarse woody habitat, or they stocked fish. Again, three potential management tools to increase diversity or increase fish productivity. And they found that the one that had the biggest impact was actually the creation of new habitat, that shallow littoral zone creation shown there in that upper left. And so they argued that it was more of an ecosystem-based management approach of restoring habitat that provided a bigger payoff than sort of a single species approach of just stocking fish. So I thought that was a really interesting result that, again, sort of sets the stage for habitat restoration and its potential power. And so in our review, we found sort of three categories of habitat restoration for coregonines. The first was improving spawning habitat. The second was improving connectivity, and the third was managing nutrients specifically for the benefit of coregonines. So the spawning habitat one is back to Bass and Waite Lake when they were trying to save the Vendace. They imposed many different sort of watershed issues to improve and to reduce sedimentation, to improve or keep that Vendace spawning habitat from being as degraded as it was. For European whitefish, many of them still occur in reservoirs where the water level is actively managed, and there typically are drawdowns during the winter and in anticipation of high spring rain and minimizing flood impacts. And so the first thing they did is they worked directly with the reservoir managers to modify their hydrological regimes to make those drawdowns not as dramatic. And the second thing they did is they created these relatively small 8 square meter plots of artificial substrates that they could essentially plant down in the relatively nearshore waters where whitefish would be spawning, and then have the ability to move those artificial substrates with eggs incubating on them down into deeper waters just before the drawdown occurred. So pretty creative, but I couldn't find any evidence that either of those were effective through personal communications and through looking at sort of subsequent studies. Coming to North America, Bear Lake has some endemic Prosopium species. However, they also were suffering from this climate change where the nearshore, some of the nearshore spawning habitat is becoming dry during dry years, and those eggs are essentially being exposed. So they essentially built 4 rocky reefs in deeper waters to increase that spawning habitat. And there's evidence that all of these species use that habitat. And then even larger scale, closer to home in the Great Lakes, 7 rock reefs were constructed in the Detroit and St. Clair River systems, totaling more than 50,000 square meters. And you can see an image there provided by Michigan Sea Grant, I believe. And the idea there is to restore fish habitat, including one of the coregonine species, lake whitefish, that's shown there. And I think it has been successful in providing more spawning habitat for these eggs, for these animals in the habitat. At the same time, Jason Fisher et al.'s paper in 2018 showed that the density of egg deposition was not any higher in these artificial reefs than what it was in the control areas, but perhaps the survival of those eggs was higher. But I don't think we know that. And so the second category then is improving connectivity. So here, here's an example in the Cedar River in Washington, where the mountain whitefish, Prosopium williamsoni, was able to exploit new upstream habitat. This was a new fishway that was created for the benefit of salmonids. However, this Prosopium species took excellent advantage of that opportunity. The only example we could find of building a fishway for coregonines specifically was for a species that is endangered in Canada, Coregonus huntsmani. Don't know how you say that. The recovery plan does include completion of a fishway so that this Atlantic whitefish species can access critical spawning habitat. And then we'll end restoring habitat with managing nutrients. So there are several examples, I think broadly, where fish and coregonines have benefited from land or water policies that are meant to improve water quality and potentially also improve benthic habitat, such as oxygen. And so several papers in North America and in Europe have sort of documented those policies that have benefited many fish species. But how many have actually been put in place specifically to improve coregonines? That was the question. And we did find a few. So for example, in Wales, there's alteration of farming practices to protect European whitefish in Lynn Tigid. Back to Bass and Waite Lake, Sewage treatment facilities were improved, so infrastructure improvements to reduce the phosphorus that was being released out and to increase oxygen and egg survival of Vindace. In Canada, the remedial action plan for Hamilton Harbor is based on the oxygen needs of Coregonus arteti of Cisco. And then lastly, there's a really good example in Minnesota where more than 107 or exactly 176 refuge lakes have been identified by Peter Jacobson and colleagues. So even they have sufficient depth and water quality right now to even with future climate scenarios for these Ciscos to persist based on the models. And you can see those refuge lakes are sort of northern Minnesota, but also a cluster around more developed regions there in the central part of the state. And so to maintain water quality in some of these refuge lakes, there's been a consortium of groups, state agencies, both fishery and water quality, conservation groups, and even local lake associations that have worked together to try to reach this goal of maintaining 75% of the lake's watershed to be forested, that is undeveloped. And that seems to be, based on some work by Peter Jacobson and others, sort of the magical number to maintain sufficiently high water quality and oxygen levels for these Cisco to maintain their sort of required water temperatures. And so far, 3 lakes have reached that target specifically with the goal of Cisco conservation. All right, so the last 2 are going to be a little bit faster. So eliminating harvest or creation of protected areas. Some of you may be more familiar with protected areas more in the marine context, where it's more commonly used and very hotly debated in nature studies or bioscience studies. What are the positives and negatives of MPAs, and how can we best make them effective to conserve marine species? In freshwater, they certainly are less commonly used. And there's a pretty cool paper by Robin Abel, sort of about 2 decades ago now. How can we unlock the potential of protected areas for freshwaters? We know, especially from the marine systems, that when these refuge areas are created, that multiple species can benefit. So again, so it could be something where maybe they're not done necessarily with coregonines in mind, but coregonines can benefit. And so the best survey among our co-authors that we could find was in Sweden, where they looked at all the different refuge areas, protected areas in Swedish lakes, and found that on average, they were relatively small areas of the lake, less than 2%, and only rarely was fishing banned in those places throughout the entire year. And when there was a species that was targeted for sort of, when they had this in mind, when they created the protecting area, typically it was trout, and it was not a Cisco or a whitefish. One exception was Lake Vättern, where a reserve has been in place since 2005, and it's relatively large. It protects at least 16% of the lake. And whitefish is one of the specific stocks that are noted there as one of the goals of that protected area. Another example we found is in the Baltic Sea, up in the sort of Bothnian Gulf region of the Baltic Sea, where in 2011, a no-take reserve, relatively large, of 147 square kilometers, was built within an even larger seasonal spawning closure area. And this was established for about 5 years, specifically to protect sea spawning Coregonus marina. And you can see here this no-take reserve in orange. You can see how the biomass built up, in that region over the time that no-take reserve was in place. But once that reserve was removed, the CPUE of Coregonus marina really was no different than sort of reference lakes or within the broader region where there was a seasonal spawning closure. So this study sort of indicated that it does provide the potential to increase local densities, but you have to maintain that reserve area in order to keep those densities high in the protected area. And then finally, what about examples of just eliminating harvest altogether in the entire lake, not just within a specific protected region? There's a new one in Lake Constance starting this year in 2024. There's a ban for 3 years on using whitefish nets for commercial fishers and for whitefish hooks for recreational fishers. And so they're closing all whitefish fisheries. And this is being implemented through the International Lake Constance Fishery Commission. And this is due to very low levels of catch for lake whitefish. This is their way of trying to maintain that population for future fisheries. Another example is in Estonia, where in Lake Pepsi, they closed the Vendace fishery from 2000 to 2006. It was reopened when environmental conditions improved. In Lake Michigan, finally, we had a short closure of bloater in 1976 and 1977, owing to concerns about the population being too low based on fishery independent and fishery dependent data. When it was reopened, however, the population reached record levels of abundance in the 1980s and 90s. As a side note, the fishery itself never recovered. That was due more to sort of changes in market practices and sort of market desires than sort of availability of the fishery, however. So the last example then is controlling invasive species. Again, broadly speaking, controlling invasive species is a challenge, even if you have the will and even if you have the resources. One of the best examples in the world, however, is right in our doorstep in the Laurentian Great Lakes, where scientists and managers have been able to exploit the life history of sea lamprey to reduce their abundances to 10% of their peak levels that once occurred back in the 1960s. And of course, had this been able to be achieved, say, 50 or so years earlier, that might have also benefited coregonines. So what are examples of controlling invasive species specifically to benefit coregonines? Not very many. And some of them have been more sort of exploratory. So for example, there's been some models back in the Adirondack around whitefish where they have sort of explored how effective it might be to use rotenone to eradicate all potential competitors and predators of invasive species before reintroducing round whitefish back into them. The best examples have been back in the UK, where in those lakes where they were trying to conserve Vendace and whitefish, they banned the use of live baits in the English Lake District, where those populations were still hanging on, to prevent the spread of invasive species. Up in Canada, with Hunt's Maneye, there's a pilot program underway now to use electrofishing to reduce the densities or to eradicate invasive piscivores that are deemed to be a threat to that Atlantic whitefish's recovery. And then again, finally, there's some planning underway to figure out in Lake Constance, given the tremendous decline of European whitefish, the concern that is due to this new combination of invasive mussels and three-spine stickleback. How they're exploring how might they increase the densities of molluscivores to reduce densities of quagga mussels. And they're also thinking about using bottom trawls at periods of high catchability, where you might be able to remove sticklebacks at a rate high enough to reduce their densities. All right, so I know I've maybe talked a little bit longer than most people do, so I'll try to wrap it up quickly with conclusions here. Despite the widespread distribution of coregonines and their historical and contemporary declines in diversity, I think we were kind of surprised that there haven't been more instances of targeted conservation or restoration strategies. You know, the best examples I shared were those that were really legally mandated, the ones in the UK for Vendace and for whitefish, and the ones in Canada for Atlantic whitefish. But of course, we're now on this new era in the Great Lakes where we have instances of voluntary restoration and reintroduction in the Great Lakes outside of the Endangered Species Act or the Species at Risk Act sort of frameworks. But in general, sort of coregonines, not surprisingly, their conservation efforts lag behind those of trout and of salmon. even in the sort of the scientific realm, we haven't sort of had conservation or restoration at front of mind, based on sort of the word cloud exercise that we undertook. One caveat to that is that some of the genetics work that has been so popular in that coregonine symposium certainly was done within the context of informing sort of conservation strategies. Again, and then just revisiting our imperfect review where we looked at across all of these case studies and the number of lakes. I think there's some real promise or potential for thinking about some of these other strategies that have been used in mostly in Europe, I would say. So translocating fish, restoring habitat, protected areas, those ones that we found some evidence of them being occurring. They certainly were not the most common, but it might be a potential way for us to think about bringing some of these strategies to the Great Lakes as we undertake this basin-wide, or as the managers have undertaken this basin-wide coregonine restoration framework. And when this framework was endorsed in 2018, I'll just note that we were thinking about different strategies beyond just stocking. So if you look here in this green box, where of course, again, just to orient everybody quickly, this is the coregonine restoration framework, where it's the fishery managers that are defining the restoration priorities and metrics of success. We have some scientific planning that ideally can inform a restoration plan that can be implemented and then evaluated, whereby those priorities could be adjusted by the fishery managers. But when you unpack sort of this restoration plan and look at the potential restoration opportunities, they include words like habitat, refugia, reintroduction, which can include not only stocking of hatchery-reared fish, but also translocations, and then regulatory mechanisms to control fishing mortality or to reduce fishing mortality on these animals. So we have the framework certainly there to sort of think about these alternative strategies, and hopefully this review helps us sort of make the contacts with others and learn more about the efforts in Europe and Asia that we might learn from as we undertake this work in the Great Lakes. Thank you all very much for your time and attention. I'm happy to take any questions.
Great. Thanks very much, Bo. We appreciate that insightful review, and it's really nice to kind of see both the North American side of things, but also the European side of things. All right, I'll just start with a question that was posed in the chat, Bo, and then we'll go to the audience and see if there's any other questions. So one second here, let me pull this down. The question is from Kevin Rath: were zebra mussels a potential factor in the littoral zone habitat restoration, which showed positive results in Germany? If we were to improve or create new littoral zone habitats in North American lakes, would zebra mussels or other invasive species negate those efforts?
Yeah, that's a good question. I don't recall them talking about the potential increasing of habitat for nuisance invasive species in that study. But that's certainly something you'd want to think about if you were to undertake, that kind of sort of effort in in North America, where we have zebra and quagga mussels that are still really problematic. I think the bigger point of that sort of study was just to think more about how maybe short-term but costly investments in habitat can pay longer-term dividends in terms of fish production, in some cases, relative to some other strategies, such as stocking that sort of can require more consistent investment every year, especially if there isn't wild reproduction of those stocked fish, which is in some cases the case.
Great. Jay Glaze, are you still on? Jay put a fairly lengthy question in the chat, but if he's willing to voice it, that would save me from reading it. Jay, are you willing to do so?
Yep, I'm here. And basically the gist of the question, Bo, is if you think that paying attention to lake and lentic habitat is finally catching up with riverine habitat, because we've recognized for decades the degradation of rivers. Probably because you put a dam on a river and the next day you see the impacts.
Right.
But lake habitat, once you get below the Secchi visibility line, it's kind of sight unseen and sight out of, or out of sight, out of mind. And I'm just wondering if you think that we're finally catching up with that.
Yeah, no, I think that's a great point, Jay. And I admit to being somebody that really didn't think much about habitat from my Great Lakes sort of job in the first decade or so in my career, in large part because I didn't really see it as limiting potentially, and maybe didn't see it as easily as I might walking along a stream or sort of observing sort of a near shore hardening of the habitat, or as you say, a dam. But I think what's changing is the video, the GoPro cameras. So now we can see, like when we put, for example, on our bottom chart, when we put a GoPro camera on there, and we could see all of a sudden how the mussels were completely carpeting the bottom of the lake in what used to be just sandy habitat. I think more and more our eyes are being opened through new technology as to how the habitat is changing in areas that were much more difficult to see sort of a decade or, a couple of decades ago. But I agree with your point. Like it's much easier to see the habitat degradation at smaller scales. And we're finally, as you say, catching up with it potentially more in the deeper lentic habitats, and recognizing, the potential power of improving habitat in some of the offshore waters of the Great Lakes. And there's been a lot of work at the lake technical committee levels and the lake committee levels and thinking about environmental objectives and what are some of the key habitat investments that where we can put some of our Great Lakes restoration dollars, for example, to enhance fishery habitat. And I'm hoping that we can, in the future, sort of make those connections more explicitly with sort of benefits to coregonine, sort of spawning habitat as well. But one of the big challenges that we continue to face for coregonine deep water or even shallow water habitat is just not knowing what that habitat really is very well, especially for some of the deep water ciscos. Again, we have some work going on to try to reduce some of that knowledge gap, but it's gonna, hopefully we'll learn a lot over the next decade or so about what could be some of the critical spawning habitat or nursery habitat for some of these coregonines so that we can effectively begin to use some of those restoration dollars in multiple ways, including habitat.
Yep, great question, Jay. Nick Mandrak added in the chat that in Canada, there's also legally mandated conservation for superior kai and whitefish species pairs in 6 inland lakes. But he noted that there's not been actions to date. I think that's through the COSEWIC process as part of the Species at Risk Act. I'll also note
one of the great things about giving this talk is that I'm alerted to things that we missed. So if anybody has some examples that we could include this, we have a paper that's been reviewed and is sort of in revision now as part of the symposia, but we can still add some additional case studies if anybody'd like to share them. So that's a really great point.
Yeah, Bo, at one point I did look into translocation, and there are some examples in Ontario lakes. I don't know if anyone from Ontario's on. I can't remember who told me about them, but I have records somewhere, so I'll dig that up for you. I don't know that you need to add it to the paper, but it'd be something of interest for the group there.
Yeah, that'd be awesome. Was that a coregonine or was that lake trout?
Cisco, from what I remember.
Wow. Okay. Now that'd be great to have. And that's kind of the value in having sort of these local experts that, definitely could have missed some of those in Canada.
Okay, any questions from the audience? Further questions? Everybody's
An-
quiet in 2024.
Andrew?
Get him in the chat. Oh, there's Nick. Go ahead, Nick.
Yeah, sorry, Andrew. I can't find my hand signal. Oh, there it is. Sorry for diving in without putting up my hand. Couldn't find the hand for a second. Just Bo, out of interest, to me, this whole question of the coregonine problem is a key component to conservation and restoration. And then Codlett and Freihoff argued there is no problem, and it's largely because of that map that you showed of the European lakes, and they just consider every species in every lake to be a different species.
Right.
Were either of those guys present at the symposium? And because I would love to hear what they would have said about conserving ciscos.
I think we need to have more conversations with them. And it's really Switzerland where they have, that's sort of they're the ones that have been the biggest splitters.
Yeah.
And I don't think there's agreement within the EU for sort of how Switzerland has been doing. I'm trying to find the slide there. Yeah.
Yeah.
And so, so Ole Seltz was there, and I had several conversations with him. Pascal is on this talk, but I didn't meet him. He wasn't there. And then, I'm trying to, the name escapes me of one of the other plenary speakers we had. That is sort of their primary advisor. I'm embarrassed for forgetting it now. Ole Seehausen, maybe. But yeah, I think it would be best to come from them. But my recollection of sort of their perspective is that this is the best way legislatively for them to protect the diversity within the lakes. And so I think they do have some genetic evidence for differentiation between the lakes, which could be real, or it could be potentially just drift from these populations being isolated long enough. But their approach of essentially, if you have 3 whitefish species in a lake, naming each of those 3 species uniquely, even if a nearby lake might also have 3 species and might have similar sort of morphological, similar morphology and similar functionality. Yeah, it's not as clear to me how similar they sort of, they might be between lakes functionally or morphologically, even
Right,
if there's some genetic differentiation. So I think extending those conversations with them in terms of how they're handling this so-called coregonine taxonomy problem would be a really valuable thing. And the other thing where I think we could learn a lot from them is with respect, the introgression question, the hybridization question, which I know is of concern in the Great Lakes. If we were to, let's just take Lake Ontario, for example, where we are reintroducing bloater. And if we wanted to increase deepwater cisco diversity even further, and, down the road, potentially, if the managers were interested in introducing kai, would reproductive isolation be maintained between bloater and kai in a new place like Lake Ontario, or would we end up sort of having hybrids and introgression between those 2 species? And they have found that eutrophication, that they've provided strong genetic evidence that eutrophication led to introgression among those whitefish species. And the persistence of some genes of now extirpated species or extinct species still persisting in some of the species that have persisted. And so understanding the characteristics that led to that introgression by talking to them and their smaller, more tractable systems might be another way to help us better understand the risk of hybridization. Well, hybridization is going to happen. I think that's not a risk, but it's the introgression, potentially what's the risk of introgression or the probability of introgression happening to a point that would be of a conservation or restoration concern.
Yeah, well, I'll just add that's, this sort of question, Nick, is one of the reasons that we took sort of more of a spatial unit type approach with respect to some of the tasks in our science planning boxes for the restoration framework, because, you know, by taking that approach, we're sort of kind of ignoring the taxonomy problem, but recognizing the local diversity at a spatial scale within each of those lakes. And that diversity could be repeated within lakes using a spatial approach as opposed to a taxonomic approach. So very good point, Nick, and it is certainly a challenge. Did
Yeah,
you want to
Andrew.
respond, Nick? And then I see Lars also is queued up.
Yeah, so, and I don't disagree with the approach, Andrew. If you sort of corner yourself if you partition all these species into separate species and then they go extinct. Legally there's no going back from a taxonomic perspective. Right, once the taxonomic species is gone, it's gone. But that doesn't mean we may not want to restore it for other reasons, and for example, the reasons that you mentioned, Andrew. Thank you, Bo and Andrew, for this discussion. It's been very interesting.
Thanks,
Thanks,
Nick. Lars?
Okay, so very interesting, Bo. I like this survey of the European part. For some reason, I like Europe. But I also like Lake Baikal. And when I was there in 2017, they were very concerned about the decline in the Omol and the coregonines there, and they were trying to build stocking programs. Did you have a chance looking at that those informations too?
Yeah. I mean, I did what I could from sort of the primary literature and the symposia proceedings and didn't find much information there. In terms of interacting with the Russian scientists, there just wasn't an opportunity given sort of political reasons. They weren't able to attend the conference, and with my status, I wasn't able to reach out to them as a US scientist to a Russian scientist. But you're right. I think this is, again, it's an imperfect review. One of the imperfections is that we didn't have as much from Russia as we would have benefited from.
Are you interested in doing that at this stage or not? Yeah, I guess my question is, do you want me to send you some contact or get some contacts from
let's talk about it offline. I can tell you more offline. Yeah.
Excellent. Thanks, Lars. Bo, I don't know what time you want to wrap up here, but there is one more question in the chat.
Yeah, I'm fine.
Okay, so Brian Weidel revisiting the notion of translocation here raises the question of challenges associated with fish health and some of those regulations. And in fact, we do have, I think there's a position from the Fish Health Committee around translocations that we might want to take a look at. But he asks sort of whether or not translocation puts us at risk of disease transfer and how you kind of reconcile that.
Yeah, I think it definitely, there is a risk there, to be sure, as we all know. And I don't know how they overcame it in the UK. I did reach out to Ian Winfield, who seems to be the biggest expert in terms of if they had an SOP that we could follow if we wanted to explore this, because they did it not only with adults, but also with eggs, or with embryos. And then also they would incubate the embryos and the hatchery, and then prior to hatch, they'd release those into the lake. So I think there's a lot of different things to look at. I don't know what life stages would have the biggest risk of introducing disease. And I'm certainly very familiar with sort of the need to protect our own hatcheries as we bring gametes, embryos into the labs. So I don't have a great answer, Brian, but I think it's more that we need to learn more about to see if this is a viable strategy for us to add to our toolbox.
All right, it appears the questions are slowing down a bit, but if I might, I would like to call on, I see a few of the fishery management folks on here and see if they have any perspectives of about the work that Bo presented in terms of looking across UK and North American lakes, and if they have any comments or feedback with respect to the information Bo presented. All right, Scott, go for it.
All right. Thanks for the invitation, Andrew. Actually, I had a lot of things rolling around in my head. I mean, this was a great talk and very complex and things that we deal with on the front line. And a lot of things that come to mind to me is the complexity of it and the irony with a lot of this, particularly with how invasive species here have turned the tables. You know, what we infer to be issues with bottlenecks in lake whitefish recruitment, for example, and the oligotrophication of Lake Michigan, for example. But you have Green Bay that just provides this perfect case scenario for this, and in the change in the gradient from south, you know, nutrient levels from south to north, and how you observed that the distribution of lake whitefish and the recruitment of lake whitefish to be consistent with that. So everything that, you know, we don't have the smoking gun, of course, but you put it all together. So if you know, you talked a lot about eutrophication in those European lakes, but frankly, that's what could be saving us in Green Bay, for example, as most of us know. So it's interesting that fine line and some of the ironies, and then with how invasive species have turned the table and opening up a lot of things that I think about is opening up areas where we have these spawning populations on the west shore of Green Bay and opening up to further habitat for them. It might be kind of one of our saving graces, but again, invasive species have really somewhat closed the door on that, because you start talking about opening up areas and breaking down dams and moving fish, and then a lot of other managers get really anxious about that when you're talking about moving these fish from ground zero of invasive species up to their pristine rivers and lakes up in, you know, northern Wisconsin and northern Lake Michigan. So I don't know, it's just kind of a stream of thought, but
Yeah.
when you've thrown all this stuff together, it's very interesting, and it shows just how complex the issue is, particularly here in the Great Lakes and Lake Michigan specifically.
No, you're exactly right, Scott. Thanks for sharing those perspectives and you know, the complexities and the challenges with whitefish, their recruitment and the fisheries, and sort of the good news in Green Bay. And you know, one of the things I kind of glossed over, but just kind of blew my mind, was Lake Constance, where they've had a long-term whitefish fishery that's had its ups and downs. It's actually recovered. I mean, the eutrophication they observed in Lake Constance was really, really high, like maybe something like you would have seen like around Fox River, Green Bay, or sort of the heyday of Western Lake Erie, but maybe even higher than that. I don't have the numbers at the top of my head, but as they've come down to more sort of maybe not even more mesotrophic numbers, that's when they've seen a recovery as more sort of oxygen has opened up and allowed them probably still have enough sort of prey to consume. But the Lake Constance story that's really surprising and just as illustrative of that complexity is that out of nowhere came this three-spine stickleback. And, you know, three-spine stickleback had been invaded sort of Lake Michigan, Lake Huron, for example, back in the 80s, I believe. And three-spine had come into Lake Constance in the 1950s and had remained at relatively low levels, kind of like what we're seeing now in Lake Michigan and Lake Huron. In fact, maybe they're even going down. I don't think when I was doing the bottom trough survey, we would catch more, you know, a three-spine stickleback in the early 2000s than what we were catching by the late teens when I'd sort of stepped off the survey. But these three-spine sticklebacks just exploded about 10 years ago when they're eating the eggs of all the whitefish, and they're competing with the whitefish, and it just sort of shows the ecological surprises that can happen with invasive species for reasons they don't completely understand, but they point to those three-spine sticklebacks as the driver of the whitefish decline, and kind of similar to the way that we're thinking about quaggas as potentially contributing to our whitefish decline. And they have quagga mussels in the lake too, in Lake Constance. So again, it's all these systems are different, but I think there are opportunities to learn from these systems that have a lot of similarities as well. So that's another sort of stream of consciousness back at you, Scott. So
Vic, go ahead.
you're on mute, Vic.
No, I say great talk. I enjoyed the global perspective. I think two things that stood out for me. One was the fact that, you know, we usually stock racings to fingerling size for stocking in the restoration effort, and the fact that there's been success, you know, just stocking fry, say, or, you know, which I know we do for other species as well, but often not very successfully. So that was interesting. And then I really want to read that study, and I think it was Germany, where they did the habitat improvement comparisons.
Mm-hmm.
And it looked like it was very near shore, things that we can do here in the Great Lakes. And some of it seemed relatively small. You know, that's the thing we deal with, is the big nature of the lakes, and you know, you wonder if you're having any effect in the small scale
Right.
stuff that we can
Right.
do, right? So
Right.
yeah, so I thought that was really interesting too, but
Okay. Yeah.
appreciate it.
Thanks, Vic. And I think you're right. Like the thinking about stocking multiple life history sages, thinking about sampling, you know, stocking some of the younger ones is something I think caught other people's ear. Like I know Chuck Bronte, I don't know if he's able to join today or not, but he was able to go to France, and I think he shared that perspective within Fish and Wildlife. And you know, I think it's certainly something to consider. It's a numbers game. You know, we could stock more potentially, but what would their survival be relative to the survival of you know, some of the older stages? So stocking eggs too. I mean, I think that's another sort of way to get around some of the pug-headedness maybe that we're seeing that could be related to rearing these animals, especially the deep water ciscos in areas that are in habitats that are completely different than what they experience in nature. You know, they're incubating maybe the same temperatures, but not at the same sort of pressure, depth level, not at the same light. And so maybe a small scale study where we actually move embryos, stock embryos. And luckily, some people in Lake Ontario, like Brian Weidel and Alex Gatch working with New York DEC are doing some of that stuff. So yeah, there's ripe with ideas, to be sure.
Thanks. Randy.
Yeah, if I could jump in, I'm going to build off what Vic said there. Great presentation, Bo. And you know, Vic kind of asked about the importance of that shallow coastal habitat. And my question is there any indication if that was tied to a particular life stage? And I'm going to frame my question, because if you look at the issues facing coregonines in the Great Lakes, both past and future, we know that all of these changes have varying effects depending on both the species, but also the life stages, the interactions of the habitat. They become critical, right? And so if you just look at Lake Whitefish and Cisco, there's been a lot of attention to some of the early life stage egg deposition over winter habitat, but it really seems like these coastal nursery habitats, these shallow, warmer bays, where these suite of environmental variables have to almost come together in a perfect soup, right? Like Scott said, some productivity, but not too much. We've seen the changes in the plankton production and composition that might favor some coregonines over others. So I'm just curious if that habitat was related to a life stage or if you've seen a pattern of that, and if, you know, especially for the Great Lakes, we are neglecting some of these coastal nursery grounds that might be more important than what we've put or given attention to in the past.
Yeah, I mean, that paper, the data are kind of messy is what you might expect with like 20 lakes and over many years. And the biggest effect was in like total CPUE, but they did break it out into different life stages. I can't recall off the top of my head if they saw similar, it wasn't larval, but they probably had juvenile. The other thing about that is these are really small systems that they were able to do this in. I mean, going back to sort of the transferability, but we can think of them almost as mesocosms, right? And I think there's some value, even in a relatively small system, where maybe you might have a handle on some things better than we're able to have a handle on the Great Lakes. The fact that they were still able to sort of see a bigger response when they improved or adjusted coastal habitat relative to just stocking fish, I think that's pretty useful to think about. So how transferable, so that result may be transferable. Maybe it's not, maybe what they're actually doing, given the size of the lake, in terms of us trying to create, you know, like they basically took a backhoe and dug out habitat to create more near shore habitat. Like they made little embayments essentially, you know, and where they created these piles of, I could try to pull out, you know, piles of, I forget what structure, vegetation structure they used. They didn't even release it. It was just a pile. So I think the idea is provocative and interesting that they got that result. And I think you're right. Like thinking about some of our critical species, thinking more about what is it that makes that critical habitat either as nursery or as egg incubation. The more we can learn about that, I think that makes a lot of sense for me to sort of spend our resources and our time to see if that is a place where we can invest and get bigger payback, to just improve the ecosystem on its own and allow it itself to become more productive, right? So
Thanks, Paul.
thank you.
Great. Well, I think we're approaching 2 o'clock, so maybe we should wrap up here, Renee and Bo, and thank everybody for the great presentation. I reposted at the bottom of the discussion the link for Ryan's talk that's upcoming, just in case people lost it there. Thanks again, Bo, for your wonderful insights and presenting that work y'all conducted for the Coregonine Symposium. Appreciate seeing that and hearing about your perspectives. Thanks, everybody.
All right, thank you.